Greetings, fellow scientists. My name is Javier Zorrilla Siller, and I'll be going to presenting to you the work I have been doing with my research team in my doctorate degree called Fast One-Pot Hydrothermal Synthesis of Hydroxyapatite. Green chemistry currently nowadays has been obtaining synthesis methods in which the substances, in specific the hazardous ones, are being reduced. Also, the reaction time and energy consumption of this. This helps a lot to also reduce the cost effective for making these materials. So in this study, it is described a single-step hydrothermal synthesis of hydroxyapatite, in which a solution prepared using calcium carbonate as a calcium source has its calcium ions detached, debounded from the molecule using EDTA as a chelating agent. After it is detached, this calcium ion, a tripolyphosphate sodium, is added as a phosphate source. This maintaining a molar ratio of calcium phosphate of 1.67 to 1, which is pretty important for making hydroxyapatite. The reaction was carried away also at pH, which is pretty important, that is 9 to 10, and in a reaction time of 20 minutes at a temperature of 160 degrees Celsius. All of this inside a reactor and atom power monowave 50 autoclave. And this produced hydroxyapatite was compared to an hydroxyapatite obtained from bovine bone, showing expected crystallographic phase of this calcium phosphate type material and structural similarities. Hydroxyapatite is a calcium phosphate with an hexagonal structure. This one has demonstrated remarkable performance in biomedicine, bone regeneration, environmental decontamination, and has properties which allows it to be used in applications such as drug delivery, biosensors, heavy metal treatment, and although this occurs naturally in a lot of natural sources such as bones, mollusks, animal bones, also um, experimental syntheses have optimized its structure for specific applications, enhancing its thermal stability, absorption, ion exchange, making relevant materials for chemistry, physics, and material science. Here, it is demonstrated also and showed the following methods for obtaining the hydrothermal hydroxyapatite and the extraction of hydroxyapatite from bovine bone. Firstly, in figure 1, it is demonstrated how from a bovine bone it is pre-crushed, then stirring chloroform for 45 minutes so the organic phase can be freed. Then this is going to let the internal phase of this pre-crushed bone a little rumble, and then it's get grounded. After it gets grounded, it is stirred in acetone for 2 hours, also for letting it free and debound more of this organic phase so it can get eliminated from the bony phase. Then heat pretreatment in an oven at 24 hours at 170 degrees Celsius is taking place. This helps to eliminate completely the organic phase that was left in the bone phase. And at the end, it is going to get synthesized for 6 hours at 750 degrees Celsius in an oven. After all of this, the hydroxyapatite powder phase is obtained from the bovine bone. In figure 2, it is explained here in this diagram how is it obtained the hydroxyapatite using the hydrothermal synthesis in this fast one pattern hydrothermal synthesis using an anthem power monopoly 50 autoclave. And the results of these both materials, these samples are here showed in this following graphics. The first one in figure 3, the FTIR spectroscopy, it showed the first one, the A and the B. The first one, which is the hydrothermal sample, and the second one, the B, this is the bovine bone sample. We can see a lot of similarities in the vibrational modes from these materials, where we can see some hydroxide functional groups, which are the ones in blue, and the ones in green, the phosphate groups, also some carbonate groups. We can see a lot of similarities between these two. The only difference is here in the A sample, which is the hydrothermal sample, which we can see that it is obtained a little water inside of the crystalline phase. That because water is involved in the synthesis reaction. Then, in the other hand, we here have the XRD diffraction, in which we also compare the hydrothermal hydroxyapatite and the bovine bone extracted hydroxyapatite. We can see that it is showed the same peaks in both of these different samples. And this is showing us that, as we were thinking, these two samples contain the same crystallographic phase, the one that corresponds to hydroxyapatite. So in conclusion, it is confirmed that the rapid hydrothermal method provided a similar FTIR spectroscopy and X-ray diffraction results to those obtained from the hydroxyapatite from bovine bone. FTIR spectroscopy showed the same vibrational mode decisions in the hydrothermal sample and in the bovine bone sample, in which the hydroxide group are gonna be placed in these wave numbers of 3,567, 66, sorry, and 604 centimeters. The carbonate at 1,400 to 1,500 and 870 centimeters. And the phosphates are gonna be located at 1,080, 1,020, 960, 630, and 562 centimeters, except for the hydrothermal sample, in which is contains some water at 1,739 centimeters. And another difference is that in the bovine bone sample, the bands of phosphate are more intense at locations of 1,080, 960, and 630 centimeters. And just a little acceleration about the X-ray diffraction peaks is that although they're similar, there is a little slight shift of 1.1 degree in the position of middle place in the hydrothermal sample. This can be due to internal compressive stresses, structural defects, and different crystal sizes, or even so, microdeformations. Thank you for your time and for your patience, your attention. This is all for today. Thank you.